Hi everyone, this is Alexis McAleer with exam mini video 4 prompt 1 of replication of DNA in a prokaryote. The initial process begins with protein binding demonstrated by the orange circle called the origin of replication. The initial binding causes the chain of DNA to unwind in a specific spot and result in a short strand of DNA. DNA in a prokaryote has to unwind before becoming a single strand to continue with replication because it's a plectonic coil. Helicase, the green triangle, enzymes attach at each end of the replication forks and play a role to break apart hydrogen. Hydrogen holds the strands together. Gyrase helps helicase in the process by positioning itself in front of helicase so it releases supercoiling first. Single-stranded binding proteins also play a role to stabilize and protect the strands while being unwound and pulled apart. They also make sure no intra-binding occurs. A lot is going on within this image here, but we are going to break it down. This is one side of the replication bubble only. Notice that each strand is now pulled apart, and replication is occurring going opposite directions on the two strands. The 3' replicates right to left, and the 5' replicates left to right. It can get confusing trying to keep track with it because it switches when DNA is synthesized. Then it reads 5' to 3'. DNA has polymerase, which helps to synthesize new DNA. This plays a key role in replication. The DNA polymerase uses help from RNA primers and RNA primase. RNA primase is an enzyme attached to another enzyme, helicase, at the replication fork. The RNA primase helps by adding a nucleotide to the sequence. They are strategically placed coming in towards the replication fork. DNA polymerase III adds nucleotides to the primer. There are arrows inside the replication bubble pointing the direction which DNA poly 3 replicates. Do you notice the orange box at the end top right of the replication bubble? That is DNA poly 1, which has exonuclease activity that takes away the RNA primers, and it puts on DNA. If something occurred to DNA pol 1, RNA primers would still take action, and that would be bad, and we would face consequences. There are a few RNA primers on the bottom strand, but the bottom strand does not keep up with the top strand, making what we call lagging strands, but the top strand contains the leading strands. In this image, you can see DNA pol 1 replaced RNA primers with DNA. There are now breaks in the bottom strand. Those breaks are called Ozaki fragments due to the lagging strands. The enzyme ligase on the bottom strand now helps with stability in the DNA.